Bismillah Rahman Rahim. Now, this is the second video on the chapter Disease and Immunity, and I've taken out some questions from other syllabuses like 0610 so that you have a practice of these questions because this is going to be examined for the first time. Now, coming to question number 3, figure 3.2 shows a drawing of a cell from the lining of the small intestine. The lumen is the space inside the intestine where food is digested. So, this is the intestine and this is the lumen. So this is the lumen of the intestine. So that means these cells are sort of like facing, this is the lumen of the intestine. So this is where the food is, and these are the cells are somewhere here like this. Now it says state the names of the three labeled structures in figure 3.2 and describe the role of each structure in the intestinal cell. So we've got two labeled structures A, B, and C. So if you look at the diagram, what is A? A are the microvilli. And what is their function? If you wrote microvilli, you did get 1 mark. And function is allows the movement of substance into the cell, or you can say increases the surface area for absorption by diffusion. Or you can say have proteins in the membrane for active transport. You see you have protein channels in the cell membrane for active transport. Then what is B? B is the rough endoplasmic reticulum. And what is this function? It's the site for protein synthesis. And C is a mitochondrion. And what does mitochondrion do? It is aerobic respiration and it provides energy. It doesn't produce energy. Please remember, you can't write produce energy. But it releases energy, or you can say provides energy for cell processes. So please do not say produce energy. Law of thermodynamics. Energy cannot be created or destroyed. So microvilli increase the surface area for absorption, or you can say increase the surface area for protein channels for active transport. B is the rough endoplasmic reticulum. Its function is a site of protein synthesis because that's where the amino acids are put together to make a protein. And C is a mitochondrion, and that is required for aerobic respiration. Now, this aerobic respiration is underlined, so you have to use this word. You can't use anything else. So it has to be aerobic respiration. B part of the question, the cholera bacterium can survive in the small intestine and the large intestine. The bacterium releases a toxin that interacts with the receptors on the surface of cells. Figure 3.3 shows the effect of the toxin. The arrow indicates the direction of movement. So you can see there's a bacterium, there's the lumen of the intestine, and then you can see the key is this is the toxin, which is this structure, which I'm going to color in green. So these are the toxins you can see here, and then you can see them here. They're going into the cell. So the toxins, and then show the effect of the arrow indicates the direction of movement. And then what do we have in the triangle? The triangle is ion X. So this is the triangle. You can see this. You can see this here, this here, this here. I'm just coloring it so you all can see it. Now it says the toxin stimulates the secretion of ion X out of the intestinal cell. State the name of ion X. Chloride ions. So chloride ions. Then it says describe the effects of the body of the secretion of ion X into the lumen of the intestine. So lumen of the intestine, this is the lumen of the intestine. So if a lot of chloride ions are going to enter the lumen of the intestine, that's not a very good thing. And that's 4 marks. So loss of water by osmosis down the water potential gradient. There are too many ions, chloride ions in the lumen of the intestine. So that's going to cause the water to move out. This is going to result in severe diarrhea. What is diarrhea? Diarrhea is the frequent passage of watery feces. So there's going to be diarrhea, there's going to be dehydration, and there's got to be loss of other ions as well and other salts as well. So this is going to be the effect of the chloride ions. Too many chloride ions in the lumen. So water will follow automatically down by osmosis. So water will also move out of the cells. And if this person is going to be frequently passing watery feces, then he is going to get dehydrated unless we put up a drip and, put in all the water, because sometimes there's also vomiting associated. So vomiting and diarrhea both will result in an extreme water loss and the body can get dehydrated. So again, wording it, loss of water by osmosis, or you can say down the water potential gradient is the same thing. If you said osmosis or down the water potential gradient, Diarrhea, another mark, dehydration, loss of ions and salts. Other ions like sodium ions will also be lost. Potassium ions will also be lost. Figure 4.1 is a diagram of Vibrio cholera. Vibrio cholera, the bacterium that causes cholera. You have to know this name, by the way. It causes cholera, and you can see there's a plasmid, there's DNA, there's ribosomes, there's a capsule, there's cell wall, there's cytoplasm, there's flagellum. Very typical bacteria. Describe two similarities and two differences between a palisade mesophyll cell and the bacterial cell shown in figure 4.1. So palisade mesophyll cell will have a cell wall, then it will have a cell membrane, it will have cytoplasm, it will have a large vacuole, it will have chloroplasts in it, it will have a large nucleus. So describe two similarities and two differences. The similarities are a whole lot of stuff. Similarities is cell wall is present in both, cell membrane in both, genetic material in both, ribosomes in both, cytoplasm in both. Differences is in the plant cell, there are no plasmids, there's no flagella, there's no capsule, there's no loop of DNA. The plant cell has chloroplast, the plant cell has mitochondria, while a bacteria does not have any mitochondria. The plant cell has a nucleus, bacteria does not have a nucleus. In the plant cell, there's a presence of a large central vacuole, and there's no such central vacuole in a bacteria. So lots of similarities and lots of differences, and I'm sure you can remember these. You'll have to actually remember these. So similarities, both have a cell wall, both have cell membrane, both have DNA, both have ribosomes, both have cytoplasm. So you can see the differences. Mesophyll cells do not have a plasmid, do not have a flagella, do not have a capsule, do not have a loop of DNA. While the mesophyll cells, what do they have? They have chloroplast. Bacteria does not have chloroplast. They have mitochondria, no mitochondria in bacteria. Nucleus present, no nucleus in bacteria. Large vacuole in mesophyll cell, no large vacuole in bacteria. Coming to part 2 of the question, explain how the cholera bacterium causes diarrhea. Now, we've done that in the previous question, but bacteria releases a toxin which causes chloride ion secretion into the small intestine. Bacteria releases a toxin. This causes chloride ion secretion into the small intestine. This reduces the water potential in the gut. So water moves in by osmosis, and water will move into the intestine and out of the cells, and that is going to cause diarrhea and dehydration and all that, which we've done in a previous question, but still I'm repeating myself. So bacteria releases a toxin causing chloride ion secretion, reducing the water potential in the gut. Osmosis occurs, water moves into the intestine and out of the cells. And this, of course, then causes diarrhea and dehydration. Then coming to the B part of the question, a scientist tested the resistance of one strain of bacteria to different antibiotics. The scientist tested solutions of 5 different antibiotics, A, B, C, D, E, A to E. She soaked a paper disc in each antibiotic solution. So we took the antibiotic solution and we took a piece of paper and we soaked it in the antibiotic solution. The paper disc with antibiotics were placed on a petri dish containing the bacteria. So already the bacteria were growing on this plate of agar jelly. And what we did is we placed these antibiotics on it. Figure 4.2 is a diagram of the appearance of the petri dish after 48 hours. The shaded areas show where bacteria grow. So this is the area where bacteria are growing. This is the shaded area. And here you can see these dots which are called colonies of bacteria. Maybe if you can Google it, you can see this. The shaded areas where the bacteria grow, the clear area shows where bacteria did not grow. So that means the antibiotics diffused out and killed the bacteria. So the largest clear area means this is where the antibiotic has been the most effective, and it has destroyed a large number of bacteria all around it. Now, A, there was very little destruction of the bacteria. Then, so the largest, the most best is E, then B, then D and C, and then A is the worst. So the shaded areas where bacteria, the clear area show where the bacteria did not grow. So area where the bacteria grow is this area, the gray area. The white area is paper disc with antibiotic. So this was the antibiotic which we placed. So this is the antibiotic A, antibiotic A, B, C, D, E. This is C, this is D, this is C, this is B. So this was the paper disc with the antibiotic. So this was the paper disc with the antibiotic. Please try to visualize this. This is the paper disc, right? And then what happened is the antibiotics must have diffused out and it destroyed or killed the bacteria around it. That is why we cannot do this test on viruses, because viruses do not grow on agar. So it's a petri dish which contained agar jelly. So we cannot grow viruses on it. Viruses are in fact non-living. Then clear areas where bacteria did not grow. Now let's see what's the question. The strain of bacteria in this investigation causes a disease, maybe cholera. Using the information in figure, explain why antibiotic E would be the most effective at treating the disease. Why? Because it has the largest clear area. That means it is so effective that it has destroyed a large number of bacteria. Largest clear area, or you can see killed the most number of bacteria, or you can see these bacteria have no resistance to antibiotic E. That means they are being destroyed. They don't have the ability to save themselves from the antibiotic. So largest clear area. Part 2, the results in figure 4.2 show that this strain of bacteria is resistant to antibiotic A. 5 years ago, a similar investigation found that the clear area for antibiotic A was the same size as antibiotic B as in figure 4.2. Explain how bacteria become resistant to. Now, I've done this in the first video, so that's why I'm not going to go into much details about it, because I feel I'm just going to be repeating myself. Natural selection, mutation, bacteria without resistance die, the resistant ones survive, and they pass on their genes, or you can see the alleles for resistance to their offsprings. So, I mean, how bacteria become resistant to antibiotics? Number one, natural selection. Number two, mutation. Bacteria with resistance survive. Surviving bacteria multiply, passing their alleles to their offsprings. Last part, describe how to minimize the risk of antibiotic B developing the same results as antibiotic A. Only take antibiotics when needed, or you should complete the course of the treatment, or you should restrict the use of antibiotic B. You should not take antibiotics in a viral disease because that's really going to not be any beneficial. Question number 5, bacteria classified in the prokaryotic kingdom. Prokaryote kingdom. State two features of animal and plant cells that are not found in prokaryotes. What are prokaryotes? They are bacteria. BP, bacteria, prokaryote. Two features of animals. Two features of animal and plant that are not found in prokaryotes. There is no nucleus. Animal and plant cells have a nucleus. So nucleus present in animal and plant cell. And there is no nucleus in bacteria. Second point, there are chromosomes. There are no chromosomes in bacteria. Then there are mitochondria. Mitochondria are present in animal and plant cells. Then endoplasmic reticulum. So endoplasmic reticulum is only that of eukaryotes, which is of the animal and plant cells. So endoplasmic reticulum present, and the last one could be vacuoles. Vacuoles are present in animal and plant cells. In plant cells, they are large central vacuoles. In animal cells, they're just small temporary vacuoles with no such vacuoles in bacteria. Then coming to the B part of the question, methicillin-resistant Staphylococcus aureus or MRSA is a type of bacterium that is resistant to some antibiotics. Figure 5.1 shows a population of bacteria may develop antibiotic resistance and how the antibiotic resistance can be passed from one strain of bacterium to another. So we have a population of pathogenic bacteria A in patient 1. Treatment with antibiotic Z started. Population of pathogenic bacteria A in patient 1 in the middle of the treatment with antibiotic Z. Population of pathogenic bacteria A in the patient 1 when treatment with antibiotic Z was stopped. Transmission of bacteria A to patient 2. Bacteria A and B in patient 2. So you see, they're showing you something. They're showing you some very important process. And they're saying bacteria of strain is this resistant to Z, then bacteria of strain A not resistant to Z, dying bacteria of strain A, bacterium of strain B resistant to Z, bacterium of strain B not resistant to Z, and the plasmid. So you can see how what is happening is that the resistant bacteria are now surviving. Now, what is the question? Explain how resistance to an antibiotic develops in a population of bacteria and spreads in the human population. Now, basically, this is the same question. I've done it earlier. Number one, resistance arises by mutation in a small number of bacteria as a random event. Antibiotics kill bacteria that do not have the mutation. Resistant bacteria have no competition. Resistant bacteria reproduce, pass on the gene, and this is natural selection. And then if the person coughs and he has the bacteria in his exhaled air, then it's going to be transmitted from one person to another. Or maybe gene transfer to other bacteria in a plasmid. So the same question, the same points that we studied in a previous question. So resistance arises by mutation in small number of bacteria. Random event. Antibiotics kill bacteria that do not have the mutation. Resistant bacteria now have no competition. Resistant bacteria reproduce, pass on the allele for resistance, or you can say the gene for resistance. This is a process of natural selection. How does it transmit it to another person? Person coughs, bacteria is exhaled and passed to another person. Or if it's passed out in the feces and those feces then contaminate a water source, then people drink from that water source, so it will be passed on to another person. Now coming to the part C, explain how the development of resistant bacteria such as MRSA can be minimized. Well, use antibiotics less often or prescribe them less often. Do not use for viral or fungal infections. Make sure people complete their course of antibiotics. Develop new antibiotics. Do not use the same antibiotic for too long, or you can say rotate antibiotics. Or you can say use combination of antibiotics. And the last one would be isolate the patients with antibiotic resistant infections so that they do not spread it to other people. So how can we minimize the development of resistant bacteria? Is very simple. Use antibiotics less often. Sometimes, you just take Panadol and 3 times a day for 2 days and your fever is gone. So you actually don't need to take an antibiotic, but you, of course, because antibiotics are freely available in Pakistan, you can just go and buy them off the counter. You can't do that in other countries. Pakistan is the only country and some other countries where you can buy antibiotics you can just go to the shop and buy antibiotics. You can't do this in London, you can't do this in New York. So we must say Pakistan is a real free country. Use antibiotics less often. Do not use in viral or fungal infections. Make sure that people complete the course. If the doctor says take this antibiotic for 5 days and after 2 days you're feeling better and you stop the antibiotic, well, you are going to develop the resistant strains of the bacteria. So make sure people complete the course of antibiotics, develop new antibiotics because now we are running out of antibiotics. Then rotate antibiotics because in certain diseases we keep on giving the same antibiotic. Like I remember in the typhoid we give an antibiotic called chloramphenicol, which is also called chloromycetin. But now we're getting resistant strains of it because normally if anybody has typhoid, we just give chloromycetin. So rotate the antibiotics. Don't give everybody chloromycetin. Give somebody something else. Give others something else. So the doctors have to be trained in that way. use a combination of antibiotics. Sometimes, like in the disease called tuberculosis, we give 3 antibiotics altogether, and we give it for 9 months. Then we isolation of patients with antibiotic resistance infections so that they don't spread it to other people. If they're coughing and spreading it to other people, well, the poor other people are going to also be suffering, and they're going to be resistant strains are going to be increasing. And this completes video 2. And thank you very much. And I will continue with a few more questions, which will go into the third video on this chapter. This is the chapter on disease and immunity. Thank you once again. And please leave your comments and please do subscribe. Thank you very much.